Hello and welcome my dear students and biochemistry learners. Welcome to this definition series in Biochemistry Part 8. In this video, I'll be informing some very simple and basic definitions related to hemoglobin. Hemoglobin. Hemoglobin with molecular weight 64,450 Daltons is a conjugated protein containing globin, the apoprotein part, and the heme, the non-protein part or the prosthetic group. Hemoglobin or Hb is a tetrameric allosteric protein with 2 alpha and 2 beta polypeptide chains. Myoglobin. Myoglobin MB is a monomeric oxygen-binding hemoprotein found in heart and skeletal muscles. It has a single polypeptide chain with 153 amino acids and has a heme moiety. Myoglobin with molecular weight 17,000 Dalton structurally resembles the individual subunits of hemoglobin molecule. Heme. It is iron-containing compound belonging to the class of compounds protoporphyrins. It is common prosthetic group present in hemoproteins like hemoglobin, myoglobin, cytochromes, some enzymes, etc. Heme contains a porphyrin molecule, namely protoporphyrin 9, with iron at its center. Protoporphyrin 9 consists of 4 pyrrole rings, to which 4 methyl, 2 propionyl and 2 vinyl groups are attached. Globin. Globin belongs to the class of proteins called globulins. In hemoglobin, it consists of 4 polypeptide chains of 2 different primary structures, alpha and beta monomeric units. Types of normal human hemoglobins. They include HbA1, HbA2, HbF, HbA1c. Let's see them one by one. HbA1. It is the common form of adult hemoglobin, about 98% of the total Hb. It is made up of 2 alpha chains with 141 amino acid residues and 2 beta chains with 146 amino acid residues. HbA2. In adults, a small fraction, about 2% of hemoglobin is known as HbA2. HbA2 is composed of 2 alpha 141 amino acid residues and 2 delta 146 amino acid residues chains. Fetal hemoglobin or HbF. Fetal hemoglobin HbF is synthesized during the fetal development and is composed of 2 alpha 141 amino acid residues and 2 gamma 146 amino acid residues chains. A little of it, less than 2%, may be present even in adults. Glycated Hb or HbA1c. Glycated Hb or HbA1c is formed by the covalent binding of glucose, glycation, to the N-terminal residue valine of beta chains of hemoglobin. Normally, HbA1c is found in low concentration, about 5% of total hemoglobin. It is increased in diabetes mellitus, which is successfully utilized for the prognosis of these patients. Oxygen dissociation curve or ODC. The binding ability of hemoglobin with oxygen at different partial pressures of oxygen or PO2 can be measured by a graphic representation known as oxygen dissociation curve ODC. It is also known as oxygen hemoglobin dissociation curve. At the partial pressure in the pulmonary alveoli, that is around 90 mmHg, the hemoglobin is 97% saturated with oxygen. In tissue capillaries with partial pressure of 40 mmHg, the Hb is 60% saturated. So physiologically, approximately 40% of O2 or oxygen is released. Cooperative binding of oxygen to hemoglobin. The binding of oxygen to one heme residue increases the affinity of remaining heme residues for oxygen. Homotropic interaction. This is called positive cooperativity. Thus, each successive addition of oxygen increases the affinity of hemoglobin to oxygen synergistically. On the other hand, release of one oxygen from one heme facilitates the release of oxygen from others. In short, there is a communication among heme groups in the hemoglobin function, also called as heme-heme interaction. T form of hemoglobin. The deoxy form of hemoglobin exists in a T or taut or tensed form. The hydrogen and ionic bonds limit the movement of monomers. Therefore, the T form of Hb has low oxygen affinity. R form of Hb. The binding of oxygen destabilizes some of the hydrogen and ionic bonds, particularly between alpha-beta dimers. This results in a relaxed form or R form of Hb, wherein the subunits move a little freely. Therefore, the R form has high oxygen affinity. The existence of hemoglobin in two forms, T and R, suitably explains the allosteric behavior of hemoglobin. BOHR effect. The binding of oxygen to hemoglobin decreases with increasing H+ ion concentration or lower pH, or when the hemoglobin is exposed to increased partial pressure of carbon dioxide. This phenomenon is known as BOHR effect. It is due to a change in the binding affinity of oxygen to hemoglobin. BOHR effect causes a shift in the oxygen dissociation curve to the right. Chloride shift. When carbon dioxide is taken up, the bicarbonate concentration within the cell increases. This would diffuse out into the plasma. Simultaneously, chloride ions from the plasma would enter in the cell to establish electrical neutrality. This is called chloride shift or the hamburger effect. When the blood reaches the lungs, the reverse reaction takes place. That is, reversal of chloride shift. Types of derivatives of hemoglobin. Hemoglobin readily combines with any gas or other substances to form derivatives of hemoglobin. Normal hemoglobin derivatives are oxy-hemoglobin, reduced hemoglobin, and carb hemoglobin. First, oxy-hemoglobin or oxy-Hb. This is formed by the combination of hemoglobin with oxygen by the process of oxygenation and not oxidation. It is dark red in color. Second, deoxy or reduced hemoglobin, deoxy-Hb. When oxygen is released from the oxy-hemoglobin, it is called reduced or deoxy-hemoglobin. It is purple in color. Let's see the difference between oxygenation and oxidation. The understanding of this is very important for hemoglobin chemistry. When carries oxygen, the Hb or hemoglobin is oxygenated. The iron atom in Hb is in ferrous state, Fe++ state. Oxidized hemoglobin is called Met-hemoglobin. The iron is in ferric state and the oxygen carrying capacity is lost. Okay. Carbaminohemoglobin or carb hemoglobin, carb Hb. It is the derivative of hemoglobin with carbon dioxide. It is blue in color. Now, abnormal hemoglobin derivatives. Abnormal hemoglobin derivatives are formed when hemoglobin combines with any toxic gas or other substances. Abnormal hemoglobin derivatives are Met-hemoglobin, Sulf-hemoglobin, Carboxyhemoglobin. First, Met-hemoglobin. It is the oxidized hemoglobin. The iron in the ferrous state is replaced by the ferric Fe3+ state. The methemoglobin with Fe3+ state is unable to bind and transport oxygen. Instead, a water molecule occupies the oxygen site in the heme of methemoglobin. Methemoglobin is dark brown in color. An increase in methemoglobin in blood, that is also called as Met-hemoglobinemia, is manifested as cyanosis. Sulf-hemoglobin. It is formed when hemoglobin is acted upon in the presence of sulfur-containing compounds like hydrogen sulfide that may arise from bacterial action in the intestine. Sulf-Hb is green in color. Carboxyhemoglobin. Hemoglobin binds with carbon monoxide to form Carboxy-Hb. Carbon monoxide is a toxic compound, an industrial pollutant that can bind with Hb in the same manner as oxygen binds. In fact, carbon monoxide has 200 times more affinity than oxygen. In such case, the hemoglobin is unsuitable for oxygen transport. Carboxyhemoglobin is cherry red in color. Now, hemoglobinopathies. Abnormalities in the primary sequence of globin chains lead to hemoglobinopathies. They include HbS, HbE, HbC, HbD, HbM. Let's see them one by one. Sickle-cell hemoglobin or HbS. It is the most common form of abnormal hemoglobins. Production of HbS is due to mutation in the beta-globin gene, which codes for beta-globin chains. In HbS, a change, a missense mutation in the single nucleotide thymine to adenine of beta-globin gene causes the formation of altered codon GUG in place of expected GAG. which leads to the incorporation of valine instead of glutamate at the 6th position in beta chain. The RBCs of people with HbS sickle. The sickled cells form small plugs in capillaries. During electrophoresis, HbS moves slowly than Hba at alkaline pH, and at acidic pH, HbS moves faster than Hba. So point mutation for HbS is beta 6, amino acid substitution is glutamic acid to valine, and codon and base substitution is A is replaced to U. So GAG is changed to GUG. Hemoglobin E or HbE. This is the most common abnormal hemoglobin after HbS. It is primarily seen in Orientals of Southeast Asia, countries like Thailand, Myanmar, Bangladesh, etc. In India, it is prevalent in West Bengal. HbE is characterized by the replacement of glutamate by lysine at the 26th position of beta chain. The HbE has similar mobility of HbA2 on electrophoresis. So point mutation is beta 26th residue. Amino acid substitution is glutamic acid to lysine, and codon and base substitution is GAG to AAG. Cooley's hemoglobin or HbC. It is characterized by substitution of glutamate by lysine in the 6th position of beta chains. The RBCs of people with HbC do not sickle. However, crystals of HbC may form within the cell. The HbC is seen mostly in the black race. The HbC is slower moving than Hba on electrophoresis at alkaline pH. So point mutation is beta 6th position. Amino acid substitution is glutamic acid to lysine. Codon and base substitution is GAG to AAG. Hemoglobin D or HbD. This is caused by the substitution of glutamine in place of glutamate in the 121st position of beta chain. Several variants of HbD are identified from different places indicated by the suffix. For example, HbD from Punjab, HbD Los Angeles, etc. HbD on electrophoresis moves along with HbS. So point substitution is beta 121. Amino acid substitution is glutamic acid to glutamine. Codon and base substitution is GAG to CAG. M-Hemoglobins or HbM. These are a group of variants where the substitution occurs in the proximal or distal histidine residues of alpha or beta chains by tyrosine. Alpha 58 histidine to tyrosine is HbM Boston. Beta 92 histidine to tyrosine replacement is HbM Hyde Park. As a result, the heme has a tendency to get oxidized to Heme, forming Met-hemoglobin. Point mutation position is proximal or distal histidine in alpha or beta chains. Amino acid substitution is histidine to tyrosine. Codon and base substitution is CAC to UAC. Thalassemias. Abnormalities in the rate of synthesis would result in thalassemias. In other words, normal globin chains in abnormal concentrations result in thalassemias. Example, beta-thalassemia. The word itself is derived from a Greek word thalassa, means sea. Mostly occurs in the regions surrounding the Mediterranean Sea. These diseases, however, are also prevalent in Central Africa, India, and the Far East. Alpha-thalassemias. Alpha-thalassemias are caused by a decreased synthesis of total absence of alpha globin chains of Hb. 4 types of alpha-thalassemias occur, which depend on the number of missing alpha globin genes. First, silent carrier. This state is due to loss of one of the 4 alpha globin genes with no physical manifestations. Number 2, alpha-thalassemia trait. It is caused by the loss of 2 genes, both from the same gene pair or one from each gene pair. Minor anemia is observed in alpha-thalassemia trait. Third, hemoglobin H disease. It is due to missing of 3 genes and it is associated with moderate anemia. Fourth, hydropus fetalis. It is the most severe form of alpha-thalassemias due to lack of all the 4 genes. The fetus usually survives until birth and then dies. Beta-thalassemias. Decreased synthesis or total lack of the formation of beta globin chain causes beta-thalassemias. The production of alpha globin chain continues to be normal, leading to the formation of a globin tetramer, all 4 alpha, that precipitates. This causes premature death of erythrocytes. There are mainly 2 types of beta-thalassemias. First is beta-thalassemia minor. This is an heterozygous state with a defect in only one of the 2 beta globin gene on chromosome 11. This disorder, also known as beta-thalassemia trait, is usually asymptomatic, since the individuals can make some amount of beta globin from the affected gene. Number 2, beta-thalassemia major. This is a homozygous state with a defect in both the genes responsible for beta globin synthesis. The infants born with beta-thalassemias major are healthy at birth, since beta globin is not synthesized during the fetal development. However, they become severely anemic and die within 1 to 2 years. Frequent blood transfusion is required for these children. This is associated with iron overload, which in turn may lead to death within 15 to 20 years. I am thankful to all these resources for making this video possible. Thank you everyone for helping me in making this video possible. Thank you friends. Thank you for watching this video. Be happy, healthy, and stay safe. And remember, biochemistry is easy. Thank you so much.